My name is Bob Langer, and I'd now like to go over my third lecture, which is Biomaterials and Biotechnology, the Development of Controlled Drug Delivery Systems, and the Foundation of Tissue Engineering. In my last lecture, I went over the fact that numerous angiogenesis inhibitors were approved by regulatory authorities and are now in clinical use, and that controlled drug delivery systems actually helped in creating the bioassays that enabled many of them to be isolated. Also, I went over the fact that one could create new smart materials like nanoparticles and even smart microchips that could be externally activated and used in the body. Now what I'd like to do is turn to some of the materials themselves and some of the issues with those. So as I mentioned in my second lecture, I worked in a hospital for a number of years. And one of the things that I was curious about as a chemical engineer was how did materials find their way into medicine? And I thought, naively, that it must be chemists and material scientists and engineers that did that. But when I looked into this, I found out that was almost never the case. It was almost always clinicians. And what they did is they would usually go to their house to find some object that resembled the organ or tissue they were trying to fix, and they'd use it in a person. For example, in the case of the artificial heart, in 1967, some of the clinicians at NIH wanted to make a heart, and they wanted something with a good flex life. And they said, well, what object has that? And they said, a lady's girdle. So they took the material in a lady's girdle and made the artificial heart out of that. And that's actually not only what was used in 1967, it's what's used today. But one of the problems is that when one starts down that path from a regulatory standpoint, it's very hard to change it. And when people designed the artificial heart, many times it hasn't worked that well, because when blood hits the surface of the artificial heart, the lady's girdle material, it could form a clot. That clot could go to the patient's brain, they got a stroke, and they may die. And yet, to me, it's not that surprising that something that was designed to be a lady's girdle material might not be the optimal blood contacting material. And this problem really pervades all of medicine. Dialysis tubing was sausage casing. Vascular graft, that's artificial blood vessel, was a surgeon in Texas going to a clothes store to see what he could sew well with. And breast implants, one of those was a lubricant, another actually was a mattress stuffing. Being a chemical engineer, I thought, well, maybe there's a different way. One of the things you learn about in chemical engineering is design. And so I thought, why couldn't we ask the question, what do we really want in a material from an engineering standpoint, chemistry standpoint, and biology standpoint, and then could we synthesize it from first principles? So we picked an example. When I started, the only material that was FDA approved that was synthetic and degradable were the polyester sutures. And they dissolve by a process we call bulk erosion, which means that as you look at the top part, it's intact, but then over time it gets spongy all the way through, and then it just breaks apart, which is okay for some drugs, but for others it could lead to bursts of drug release, and that could be fatal if you had a possibly dangerous drug like an anti-cancer drug. So we said, and others have said, what you really want in a polymer to degrade is not bulk erosion, but surface erosion. That would be layer by layer erosion. And if that could happen, kind of the way a bar of soap might dissolve, then you wouldn't have this problem of dose dumping. The challenge was, how could you do that? How could you create a polymer to do this? So we went through a very detailed engineering design analysis to try to figure that out. We'd start out with different design questions, like what should cause the polymer degradation? Should it be enzymes? Should it be water or something else? Our thinking is, well, everybody may have different enzyme levels, but everybody has excess water. So let's build into the polymer the ability to be degraded by water as a first step. And then what we did is we figured out what the right building blocks are to make those polymers that would keep water out, because you want to keep water confined to the surface. And then we tried to figure out what would be the right chemical bonds that would break apart in the right way, and we came up with the anhydride bond. And then we tried to figure out what would be the specific units in the polymer that would be safe in the body, and ultimately we came up with this polymer. It's what's called a copolymer. There's two units to it. PCPP, that's carboxyphenoxypropane, and sebacic acid. And our thinking was that by changing the ratios of these, we could make the polymer last for different times. And if we use this anhydride bond to connect everything, then we might be able to get the surface erosion. We made some of these. We synthesized these polymers. It's a good deal of work, but we synthesized them. And what you see is they actually did come quite close to the surface erosion. But if you change the ratio of one to the other, the 79, 55, and 15, and 0, all referring to the amount of subbasic acid, what happens is you can make these last for almost any length of time. You can go from the 79% one, it lasts for about 2 weeks. The 0% one will last for 3 or 4 years. So you could simply dial in the ratio of these and make them last for almost any length of time you want. Well, one of the things that I always want to try to do is not just write the paper, but to try to see if we can use these materials we create to do something useful. And in 1985, Henry Brehm, a young neurosurgeon at Johns Hopkins, came to visit me and wanted to see if we might be able to help him come up with a different way to treat brain cancer. Henry is now chairman of neurosurgery at Johns Hopkins. But just briefly, these were some of the statistics at the time. With glioblastoma multiforme, it was a uniformly fatal disease. The mean lifespan, regardless of how you treat it, was generally less than a year. The drug that people used at that time, the 1980s, was this one, BCNU. It's a effective but very toxic drug. And what Henry and I talked about was this idea. Rather than give it intravenously, which was always what was done before, could we introduce this paradigm of what I'll call local chemotherapy? Could we allow the neurosurgeon, like Dr. Brehm, to operate on the patient, to remove as much of the tumor as he could, but before he closes the patient, could he line the surgical cavity with little wafers containing this drug and polymer? Now, this polymer, the drug, I should say, normally lasts just for 12 minutes. But if you put it in the polymer, it's protected. It'll last as long as the polymer lasts. So the neurosurgeons like Dr. Brehm, they wanted it to last for a month. And we could do that by changing the chemistry. So basically what we were able to do is make these wafers, make it last for a month. And also what was important to the neurosurgeons is they're putting the wafers in the brain. So they're exposing only the cells largely they wanted to the drug. And the rest of the body is really spared these high concentrations of chemotherapy. So so that was the idea. And but what happened is that whenever you're in academia, professor, you have to raise money. And so what we would do to try to raise money to move this forward, first to create the new materials and everything, is I'd write grants, and I'd write them to like the National Institutes of Health or other places, and then they'd have professors at other universities review them and say what they thought. And we did terribly. We did very, very badly. When I first wrote the grants, the reviewers, the chemists said, well, we'll never be able to synthesize the polymers. But I had a very good graduate student at the time named Howie Rosen. Howie later became president of the Alza Corporation, a $12 billion corporation, and also has been elected to the National Academy of Engineering, and he synthesized the polymers. So we sent the grant back, and the reviewer said, well, still shouldn't fund it, even if you can synthesize them. The polymers are they have these anhydride bonds, they'll react with whatever drug you put in. But I had another couple of postdocs, Bob Linhart, who's now Constellation Professor of Chemistry at RPI, and Cam Leung, who's a James Duke Professor of Bioengineering at Duke University, also has elected to the National Academy of Engineering, and they showed there was no reaction. So we sent it back again, and the reviewer said, well, okay, that's not a problem, but these polymers are low molecular weight, they're fragile, they'll break in the body. But I had another couple of postdocs, Edith Mathiowicz, she's now full professor of bioengineering at Brown University. She's been elected to the National Academy of Inventors, and Avi Dome, who later became chairman of medicinal chemistry at Hebrew University. And they made polymers that were very strong, high molecular weight polymers, and wouldn't break. So then sent it back again. The reviewer said, well, new materials are certainly going to be toxic. But I had another graduate student, Cato Lorenzen. Cato later became Dean of Medicine at the University of Connecticut, and he's been elected to actually both the Institute of Medicine of the National Academy of Sciences as well as the National Academy of Engineering, and he showed that they were very, very safe. Anyhow, this kept going on and on until 1996 when the FDA approved the treatment. It was actually the first time in over 20 years they approved a new treatment for brain cancer, and the first time they ever approved this idea of polymer-based chemotherapy for cancer. You can probably tell from the way I'm speaking that I'm very proud of how well all the graduate students and postdocs did. They, became chair people of departments, presidents of large corporations, received all kinds of honors, whereas the reviewers, they haven't done that well. Now, I'd like to actually show what the operation looks like, but these are going to be fairly bloody slides, so people shouldn't look if they don't want to. But this is going to be a little wafer going into the human brain, and you can see that here. It's the white part, and usually they put 6 or 7 in and close up the brain. So I should point out that it's very hard to get good advice when I give a talk, but my wife once came to one of the talks I gave, and I asked her about it. And this was a talk to a group of engineers at MIT, and she told me that I had left those 2 bloody slides on for 10 minutes explaining all the details, and unfortunately all of the engineers got, I guess, very ill, and I should have noticed that. So now I don't leave them on very long when I talk to engineers, so I apologize for that. At any rate, this was some of the clinical data. It was published in Neurosurgery and from European clinical trials. It's what's called a phase 3 clinical trial. And what you see is at the end of at the end of 2 years, you do see significantly increased survival. It's not a cure by any means, but what's been exciting is that now for patients who have sometimes localized tumors, this treatment has been approved by the FDA. It's been used in over 30 countries for the last 18 years. And it created a new paradigm for how one would think about local chemotherapy. Not only might you use it in cancer, and people are studying it in other kinds of cancer, but you might also use it in other diseases. In fact, one of my other former graduate students, Elazar Edelman, who's now a professor at Harvard and MIT, as well as a number of companies like Boston Scientific, have used these ideas in the area of drug-eluting stents. And that's been a huge area in interventional cardiology. Today, if somebody has heart disease, one of the things that are often done is to prop open the blood vessel by putting a stent in. It's like a Chinese finger puzzle. But about half the time, what happens is it closes off because of smooth muscle cell proliferation. And now what's done is to coat these stents with a polymer that locally delivers a drug like another anti-cancer drug like Taxol, and that prevents that proliferation. And these are used in about 1 million patients every year and have had very profound effects. Now what I'd like to do is go to the second part of the talk, where I'd like to talk to you about using materials to create new tissues and organs by combining materials with cells. Here I've worked very closely with Jay Vacanti. Jay is head of the pediatric surgery program at Massachusetts General Hospital. And he and I have been working on this for now over 30 years. And the reason this came about is he would see patients who had liver failure, like this little boy, who were dying, and there was no way to treat them other than a transplant. And there weren't nearly enough transplants. And so he and I started talking about this and asked, could we come up with a way to maybe use the patient's own cells or a relative's cells or someone's cells combined with materials to create new tissues and organs? The specific idea we had is shown here. This was from a paper we wrote in Science many years ago. And the idea was you could take these cell types, you see osteoblasts, which are bone cells, or chondrocytes, which are cartilage cells, hepatocytes, which are liver cells, and so forth, and you dissociate them. Today, you might also consider using stem cells and converting them to one of these. But if you take these cells and inject them at random in the body, not much happens. But the cells are smart, and people, for example, at Berkeley have shown that you can take mammary epithelial cells and put them close together, and when you do that, they're smart enough to actually make acini and make milk. And our theory was that if we could create the right kind of polymer and biodegradable scaffold, and the cells would be close enough together, and they were grown under the right in vitro tissue culture conditions, what we call bioreactors, maybe we could make a new tissue and ultimately put it in the body. There are a number of components to this, as one sees on the slide. First component is having the right materials, and we would generally use degradable materials that had been shown to be safe in people, and in many cases, we synthesize new materials ourselves. We might then convert them to fibers, where we could put cells on, like you can see in the scanning electron micrograph. But also, the way this field is going, we also thought that you might someday be able to even use techniques like CAD-CAM techniques. Computer-aided design is what I basically mean. And just to illustrate that, This is work that Prasad Shastri, who was a postdoc with us, did. He basically was thinking about making new structures like a nose, which you see here. And so he basically designed a nose with a foaming technique. You could also use 3D printing. Other ones of my former postdocs, like Linda Griffith, have done things like that. She's a professor at MIT now. And the idea is that you could make basically a nose that's 98% porous, but it's made of a polymer, polylactic glycolic acid. And you could make this nose into any shape. So I've just speculated 30 or 40 years from now, maybe there'll be a computer printout that somebody could pick when they're going to a plastic surgeon whatever nose shape they want. So they could have a regular nose shape, or maybe they'd want an upturned nose shape, which wouldn't be hard. You just take a little bit of this off. You could even give a hook nose shape. I mean, probably nobody would want that, but you could do it. And then maybe you take the patient's own cells and put that on the scaffold. And so these are just some of the examples. So I thought I would go through a few examples just to illustrate some of the challenges and some of what we and others do. So let's say you want to make a new blood vessel. That's been very challenging. There's not been a way to make small diameter blood vessels. So one of our students, David Mooney, who is now a professor at Harvard, made these little tubes out of a polymer that are also about 97, 98% porous. And then Jinming Gao, another postdoc, he's now a professor at Texas Southwestern, modified polyglycolic acid, PGA, so that you could get a high attachment density of what are called smooth muscle cells. And the person who led this project was Laura Nicholson. She was a fellow with us and now is a full professor at Yale. And her idea was, nobody had ever been able to make a blood vessel before, and we and others had tried making these to try to grow them. But the way you actually culture something is important. Normally, when people grow cells, it's sitting in a Petri dish that's stationary. Maybe there's a little bit of movement. But what Laura said, that's not what happens in the body. In the body, a blood vessel doesn't just stay there. It's actually hooked up to a pulsatile pump, your heart. So she said, to get this to work, we're going to need to make a bioreactor that really mimics that, and so would create what we call pulsatile radial stress. So she did that. She figured out the right medium, had the pulsatile pump, and over a several-month period would pump that media in this pulsatile fashion, 165 beats per minute, and try to make blood vessels. And she was able to do that. This was published in Science, and made these tiny little blood vessels. And when she characterized them, they were very, very similar to regular blood vessels. 50% collagen. Their rupture strengths are greater than 2,000 millimeters of mercury. You can suture them in, and they're very strong. And they show the same pharmacology as a regular blood vessel. We worked with Bill Abbott to try to put these in pigs, which is considered the best model for blood vessels. And here you see an angiogram where the blood vessels are opened months later. And Laura actually has taken this work a lot further. She actually started a company on this, and actually they're in multiple clinical trials, actually using a variation of this, a decellularized construct, meaning she's taken the cells off after she's made it. And so that's now been used in a number of patients. Now I want to move on to cartilage as another tissue. And this work was done with Lisa Freed, Gordana Vanyak, Chuck Vacanti, and Jay Vacanti. And here what we did is we took nude mice and basically mimicked what might someday happen in a person. So what happened here is that you could take the cartilage and you can take it from an animal and redo it. And if you look at the animal on the right, we've redone his skull. If you go to the next set of animals and you looked at the animal on your left, we've redone his cheek. If you open the animal up and look at him, it's pure white glistening cartilage. And actually, biochemically, it looks like cartilage, but it's still not of a good enough mechanical strength that you can help people, at this point at least in our research, that if they had a weight-bearing injury, that you could do anything. So, but it is able to help people with various cosmetic issues. And so we actually have been approached, have worked with the Army, United States Armed Forces Institute for Regenerative Medicine, and they have patients that come back from like Iraq or Afghanistan, say, without certain body parts, like an ear. So Linda Griffith, one of our former postdocs I mentioned, and she's now a professor at MIT, actually made a scaffold in the form of an ear. You see that on the top, and on the bottom, you see a scanning electron micrograph of it, and you actually see the cells and the matrix growing, and over time you'll see more cells and matrix growing, and the fibers that you see, they'll disappear, and you'll actually get an ear. And in fact, this has not been put on patients, but it has been put on animals, and it's shown to work. And in fact, Jay, my colleague, has even put some of these systems on human beings in what are called physician-sponsored INDs. Here's a 12-year-old boy at the time, and if you look at him, he's got no chest covering his heart. But he, like other 12-year-olds, likes to play baseball. But you can imagine if he ever got hit in the chest with a baseball, he could die. So actually, Jay operated on him. We made a scaffold for him, and Jay created a whole new chest for him, and he's doing fine. Also, we licensed the technology that we developed to different companies, and some of them have now made artificial skin for burn victims. And let me just show you that. Here, for example, is a 2-year-old boy who's very badly burned. You can create a product which has now been done, and it's actually approved by the FDA and been used in many patients. But the idea is that you have a polymer scaffold. You can put on neonatal skin fibroblasts, and you can actually cryopreserve these. But then you come back and you can put it in the child at the time of injury, like this. We'll come back 3 weeks later. He looks better. And 6 months later, he's pretty much healed. So these have been approved now by the FDA for patients who have been burned and patients who have diabetic skin ulcers. And the final example I wanted to give you, which is very early, but is could you someday even help people who have paralysis, spinal cord repair? And this was a project was led, it started in our lab by a woman named Erin Levick, who's now a professor at Case Western Reserve. And this was done in collaboration with Ted Tang, who's a neurosurgeon, and Evan Snyder, who's a neuronal stem cell expert at the Burnham. And basically the idea was, could we make a scaffold that would mimic the gray and white matter of the spinal cord and have an outer part that would be sort of microfabricated or nanofabricated in a certain way to provide axonal guidance, and an inner part where we could create pores where we could put neuronal stem cells that we got from Evan. And just to show some pictures, on the top left-hand panel, you see the scaffold macroscopically. Next to that, you see a scanning electron micrograph that shows you the pores. And you see another vision of that right next to it. Below it, you see a scanning electron micrograph of the outer part that Erin created. And notice the nano patterning to aid in the axonal guidance. And finally, this on your, here is the experiment. Basically, you take the spinal cord, you remove a portion of it, and then you do one of 4 things in our studies. We basically did nothing, that's a sham operation, or you put cells in, that's the second set of controls, the scaffold by itself, that's third, and the scaffold cell combination, which would be the experiment. And these animals, Erin and Ted followed them for over 400 days and really did behavioral studies and track tracing and other things. So let me just show some of the results. So first, I'll just show a typical control animal. And notice that he's not able to move support his weight very well on the backside. And as you'll see, the paws are splayed in a rather awkward fashion. So he'd get what's called a BBB score of about 5 out of a possible 20. This is done 100 days after the start of the experiment. And we'll just look at this for a little while longer. Now, if we go to a typical treated animal, mean of the treated group, this is not a cure by any means, but he is able to bear his own weight. And notice how the paws are splayed in a much more normal way. You'll see this more clearly in a second. And he gets a BBB score of about 14 out of 20. Like I said, it's not a cure, but it's an improvement. There are the paws. And so we basically did 52 animals, 13 in each group, and got good data with both the cell polymer scaffold and the scaffold actually by itself. Then this went to monkeys, and African green monkeys, and this was done by Eric Woodard and John Slotkin and In Vivo Therapeutics. And here's the monkeys, and they're put on a treadmill test. And what happens is, in the controls, they're not able to move the one leg that's been injured, as you can see here. But when you give the scaffold or scaffold cells, they do much better. It's not, again, not a cure, but it's a significant improvement. And what's happened now is, based on this, the FDA actually has given the go-ahead for start of human clinical trials at a number of hospitals around the United States. So we'll learn more about what happens in humans, I believe, in the next year. So in summary, what I've tried to go over in this lecture is how one can create new materials to solve different medical problems, and how one can create materials and combine them with cells to maybe someday make new tissues and organs. All this work in all these slides really was made possible by the terrific funding agencies that have been very kind to us, like the National Institutes of Health, the National Science Foundation, the Gates Foundation, and other foundations, different companies. And most of all, really, it's been the terrific work of really a wonderful group of students and postdocs at MIT and Children's Hospital over the years that have made this possible. Thank you very much for having me.